So for today's session, it's my pleasure to introduce the moderator, Dr. Saman Maleki. Dr. Maleki is an assistant professor in the departments of oncology, pathology, and laboratory medicine and medical biophysics at Western University. He's also a translational immuno-oncology scientist at the Lawson Health Research Institute and at the OICR. Dr. Maleki has pioneered several phase 1 clinical trials in Canada that combined fecal microbiota transplant plus immunotherapy in melanoma, renal cell carcinoma, non-small cell lung cancer, and pancreatic cancer patients. Dr. Maleki's academic research focuses on sensitizing hard-to-treat cancers to immunotherapy via tumor vaccine strategies targeting DNA repair pathways in cancer cells or modifying the gut microbiome in the host. Saman, thank you so much for moderating the session. I'll turn it over to you now.
Thank you very much, Jay, and I would like to welcome everybody to this OICR Monday session. So I'm just going to share one slide that I have before I introduce the speaker. So the topic of the day today is how to transform cold tumors or immune cold tumors to immune hot. And I think for that, for us to understand, it's important for us to understand what is it that we really define as hot and cold tumors. And this is a bit of a complex figure, but if you bear with me, what you see here is that on the x-axis, we have the continuum of tumors that they have low tumor mutation burden on the left, and you go to the right side where you have the tumors with high tumor mutation burden, such as melanomas, non-small cell lung cancer, or mismatch repair deficient colorectal cancer, for example. And on the y-axis, you have the continuum of tumors that they are what we call T-cell inflamed phenotype. This is the immune phenotype of the tumor. And as you go bottom of the figure, you see the tumors that they are immunologically cold tumors. So in this quadrant, we have on this side of the quadrant, we have tumors that they are defined as high tumor mutation burden cancers. And when you're looking at the immune phenotype of these tumors, you see a lot of T-cell infiltration, activation of interferon signaling, and all that. And you have tumors such as I said, mismatch repair deficient colorectal cancers, or a lot of melanomas and non-small cell lung cancers. And these are the tumors that generally respond better to checkpoint inhibitor therapy, and we have the most success in the clinic with them. On the bottom of that quadrant, on this lower quadrant here, you have tumors that they have high tumor mutation burden, but when you're looking at those tumors for one reason or the other, for example, like we know that maybe beta-catenin activation, the tumor looks an immune excluded, has an immune excluded phenotype, and they don't necessarily respond as well as what we had in the previous quadrant. This could be the difference between one melanoma patient responding good to immune checkpoint inhibitors and another melanoma patient with the similar mutations, they don't really respond. On this side, you have tumors that they don't necessarily have a higher tumor mutation burden, not a lot of the antigens to be recognized by T cells, but they do have a good immune phenotype. And that could be RCC, and that's like renal cancer. And that's because they have a lot of indels, for example, and sometimes we know that we have approval for of immunotherapy for those. And you have cancers like Merkel cell carcinoma that they could be virally induced or because of UV, and there is a good immune phenotype there as well. This is, I guess, the biggest challenge we face in immunotherapy, and these are cancers like pancreatic cancer, prostate, a lot of childhood cancers. These are the tumors that they're true immune cold cancers, and they are excluded from a lot of immune cells, so they have a cold immune phenotype. So we have an incredible lineup of speakers today that will tell us different strategies that you're really shifting an immune phenotype of a tumor by using maybe microbial consortia or blocking DNA repair pathways to shift their immune phenotype and push them on this side of the quadrant so you can effectively treat them better with immune checkpoint inhibitors. So our first speaker today is Megan Hong. Megan is a grad student in my laboratory, and she's in the Department of Pathology and Laboratory Medicine at Western University. And she will be talking about how DNA mismatch repair deficiency in tumors can affect its response to immune checkpoint inhibitors. And that is in the adaptive oncology perspective that we're going to have in the talk. Megan, please go ahead and share your slides.
Are my slides showing okay?
Yes.
So thank you for that introduction, and thank you for giving me this opportunity to talk a bit about my work. So as Dr. Maleki mentioned, I will be talking about how tumors can be sensitized to immune checkpoint inhibitors by inducing DNA mismatch repair deficiency. So to start off with, anti-tumor immunity refers to innate and adaptive immune responses that facilitate tumor control. And T cells are a major player in this, as they have the ability to recognize cancer cells from healthy cells and eliminate them. Now, in order to turn a cold tumor hot, one of the most important parts to that is being able to increase T cell activation, which would lead to their expansion and infiltration into the tumor. Now, in order for a T cell to be activated, a couple of things must happen. So T cells must first be primed to recognize a specific antigen. So cancer cells can produce antigens where these antigens can be presented to T cells when they are presented on an MHC molecule that is expressed on antigen-presenting cells such as dendritic cells, to which then T cell receptors on T cells can then engage with. But in order to become fully activated, T cells must also receive co-stimulatory signaling through CD28 engaging with CD80 and 86. So when these two things happen, these T cells are then ready to go and recognize and eliminate cancer cells. Now, tumors can have or acquire several mechanisms to evade or suppress these anti-tumor immune responses. For example, if they lack the production of tumor-specific or tumor-associated antigens, the lack of these antigens would not allow for T cells to be activated or primed for that can specifically recognize the tumor. So in a sense, tumors become invisible to the immune system. Now, even if T cells become activated and are present within the tumor, chronic stimulation by antigens within the tumor can cause T cells to upregulate immune checkpoint molecules such as PD-1 and CTLA-4, where when these immune checkpoint molecules engage with their respective ligands, PD-L1 and CD80, this can inhibit T cell activation or function. So with these mechanisms of evasion or suppression of anti-tumor immune responses, you basically eliminate T cells that can recognize and eliminate cancer cells. Now, tumors can become visible to the immune system if they can produce these tumor-specific or tumor-associated antigens. And tumor-specific antigens are produced as a result of DNA mutations. So in a cancer cell, they can have impaired DNA repair machinery, and this can cause an increase in tumor mutation burden. And some of these mutations can lead to the production of mutated antigens that are recognized as foreign, and these are known as neoantigens. So these neoantigens can be be used to prime and activate for tumor-specific T cells. So today I will be focused on the DNA mismatch repair pathway that corrects for mismatch base pairs that occur during DNA replication. Now, a clinically proven way to boost anti-tumor immune responses is through the use of immune checkpoint inhibitors. So immune checkpoint inhibitors are a class of immunotherapeutics that inhibit the interaction between immune checkpoint molecules and the respective ligand. So they ultimately remove the brakes off the immune system to boost those anti-tumor immune responses through increased T cell activation. So PD-1, PD-L1, and anti-CTLA-4 are the three most common immune checkpoint inhibitors being used clinically. Now, the largest clinical challenge right now is that although a large percentage of patients are eligible for immune checkpoint inhibitors, only a small fraction of patients have a durable response to these immunotherapeutics. So there's a lot of research going on to try and either develop next generation immunotherapeutics or develop strategies to improve the efficacy of existing immunotherapeutics. And there are a couple of factors that contribute to a tumor's sensitivity to immune checkpoint inhibitors, where if we look at tumor mutation burden for cancers such as melanoma or microsatellite unstable colorectal cancer, these patients tend to have a pretty good response to immune checkpoint inhibitors. Whereas for patients such as in pancreatic cancer that have low tumor mutation burden, these patients tend to not respond well to immune checkpoint inhibitors. For a majority of cancers, the tumors are often mismatch repair proficient, which means they have an intact mismatch repair pathway, so they have low tumor mutation burden, they're microsatellite stable, and as a result, they're immunologically cold. Whereas for tumors that have are mismatch repair deficient, they have an impaired DNA mismatch repair pathway, so they have a higher tumor mutation burden and they're microsatellite unstable. And as a result, they're more immunologically hot. And these are the types of tumors that tend to respond well to immune checkpoint inhibitors. And that could be seen with sporadic colorectal cancer, where about 15 to 20% of patients can be categorized as mismatch repair deficient, where with the treatment with anti-PD-1, the overall survival in these mismatch repair deficient patients tend to be much higher compared to mismatch repair proficient patients. So given what is seen in colorectal cancer, this kind of gave us the idea where perhaps we can target the DNA mismatch repair pathway in mismatch repair proficient tumors to sensitize them to immune checkpoint inhibitors. So using the RNA-seq data that is available in the TCGA database, we looked at the expression of several mismatch repair genes in colorectal and stomach adenocarcinoma samples. We found that MLH1 was consistently downregulated in microsatellite unstable samples compared to microsatellite stable and normal healthy samples. Whereas for the other mismatch repair genes, the expression of those genes were not as affected among the microsatellite unstable samples. So given that MLH1 is commonly downregulated in the microsatellite unstable cancers, we wanted to see if inactivating MLH1 in neuroblastoma can sensitize them to immune checkpoint inhibitors. And neuroblastoma is a pediatric cancer that can be categorized as immunologically cold. It has low tumor mutation burden and it's refractory towards immune checkpoint inhibitors. So when we used the mouse neuroblastoma cell line, Neuro2A, and we inactivated MLH1 by knocking out the MLH1 gene with CRISPR-Cas9. So here on out, I will be referring to the cells that failed to have the MLH1 knockout as mismatch repair proficient, or pMMR, and those that have the MLH1 knockout as induced mismatch repair deficient, or IDMMR. So when we established these cells, we allowed these cells to grow in culture for about 8 weeks to allow for those mutations to accumulate before we use them for animal experiments. And when we grew these tumors in immunodeficient mice that lacked an adaptive immune system, we found that both these tumors grew at a similar rate. So when we grew these tumors in immunocompetent syngeneic mice, we found that the induced mismatch repair deficient tumors grew significantly slower compared to the mismatch repair proficient tumors. And this difference seen here can be said to be mediated by immune responses based on what we see in the immunodeficient mice. So when we looked at the immune cell composition within these tumors, we found that this induction of mismatch repair deficiency did cause these tumors to become more immunologically hot relative to the mismatch repair proficient tumors, where we did see an increase in both CD8 and CD4 T cells. And among the CD8 T cells within the induced mismatch repair deficient tumors, these CD8 T cells also exhibited increased cytotoxic potential that was shown by an increased presence of the CD107 expressing CD8 T cells. But most importantly, This induction of mismatch repair deficiency did not cause these CD8 T cells to increase the expression of exhaustion markers that are often indicative of reduced T cell function. So this induction of mismatch repair deficiency was able to increase T cells and also increase the effector activity of these CD8 T cells without inducing exhaustion. To next look at if induced mismatch repair deficiency can sensitize tumors to immune checkpoint inhibitors, we treated mice that had the mismatch repair proficient or the mismatch repair deficient tumors with anti-PD-1. And we found that in both models, anti-PD-1 did not have an effect in either of these tumors. However, if we did the same experiment, but with anti-CTLA-4, we found that anti-CTLA-4 had no effect against these mismatch repair proficient tumors compared to the control group. Whereas in the mismatch repair deficient group, anti-CTLA-4 was able to significantly reduce tumor burden compared to the control group, where about 60% of mice are cured at the endpoint. And this was shown to be dependent on the presence of CD4 T cells, where if we were to deplete CD4 T cells and then treat with anti-CTLA-4, the therapeutic effect of anti-CTLA-4 is completely lost. And this just further emphasizes the importance of the presence of T cells in the tumor in terms of response to immune checkpoint inhibitors. We then wanted to see if the combination of anti-PD-1 and anti-CTLA-4 in combination had any additive or synergistic therapeutic benefit compared to monotherapy. So we took these mice that had the induced mismatch repair deficient tumors and treated them with anti-PD-1 or anti-CTLA-4 as a monotherapy or in combination, and found that the combination of these immune checkpoint inhibitors did not have any additional therapeutic benefit when compared to anti-CTLA-4 alone when it came to overall survival. But what was most interesting about this research was the immune memory response that was generated, where if we took the mice that had the induced mismatch repair deficient tumors that were cured with anti-CTLA-4 and re-challenged them with the induced mismatch repair deficient cells, 100% of these mice were able to reject tumor establishment compared to naive mice that were challenged for the first time. And we also tried to challenge them with the mismatch repair proficient cells. And again, these mice were still able to completely reject the establishment of these tumors. So this induction of mismatch repair deficiency in combination with anti-CTLA-4 not only generated a pretty robust anti-tumor immune memory response against these induced mismatch repair deficient cells, this memory response was quite broad as it was able to act on these mismatch repair proficient cells that are less immunogenic. And this can be explained by, possibly explained by epitope spreading, where the combination effect of induced mismatch repair deficiency and anti-CTLA-4 may be allowing T cells that recognize more rare or cryptic antigens to become activated that can act on these mismatch repair proficient cells. And this is very important in the context of preventing cancer reoccurrence that is common in high-risk neuroblastoma patients and eliminating metastatic lesions that are often less immunogenic compared to the primary tumors. So in summary, I show that targeting the mismatch repair pathway can trigger an anti-tumor immune response that can be enhanced with immune modulators. And specifically in our work, we show that this induction of mismatch repair deficiency by inactivating MLH1 was able to sensitize neuroblastoma to anti-CTLA-4 and was able to generate a very robust and broad anti-tumor immune memory response. And ultimately, we have the goal of possibly developing a small molecule inhibitor to target mismatch repair proteins that can be used in combination with immune checkpoint inhibitors to improve their therapeutic efficacy. So to wrap this up, I would like to thank my supervisor as well as members in my lab and our collaborators and funding sources that supported this research. Thanks for listening.
Okay, thank you. Thank you very much, Megan. So now we are going to move to the second part of the event. So it's an exciting collaboration between a medical oncologist and an infectious disease doctor. So I'm going to introduce both of them. First, I start with Dr. Bryan Coburn. Dr. Coburn is a clinician scientist in infectious diseases at University Health Network with a laboratory focus on human trials of microbiome targeted therapies in infectious and non-infectious diseases. And he is collaborating in this work with Dr. Anna Spreafico. Dr. Spreafico is an assistant professor at the University of Toronto and staff medical oncologist and clinician investigator in the Division of Medical Oncology and Hematology at Princess Margaret Cancer Center and at University Health Network. She's previously trained, her previous training includes a PhD in translational research in the program for evaluation of targeted therapies at the University of Colorado, USA, and a subspecialty fellowship in head and neck cancer and experimental therapeutics in the RAS phase 1 program at the Princess Margaret Cancer Center. Anna is the head and neck medical oncology disease site lead and clinician investigator within the tumor immunotherapy program. She also serves as the director of the phase 1 drug development fellowship at the Princess Margaret Cancer Center. I think they are going to talk on the clinical translation perspective, and the title is The Use of Microbial Ecosystem Therapeutics, a group of 30 bacterial species to boost the immune system, which is going to be a very different perspective from the first talk that you heard. Please go ahead.
Okay, excellent. Okay. Well, thank you very much. It's a pleasure to be here with you. Thank you for inviting us. Brian and I will be basically working together through this presentation. I'll start first, and then I'll just move the slide for Brian moving forward. So thank you again. So just give you a brief introduction. We heard already one of the ways to kind of target the immune system and increase immune response is via microbiome manipulation. Over the past years, the role of the microbiome has definitely raised an increased interest in the cancer field, particularly mechanisms by which we can therapeutically manipulate the intestinal microbiome to improve immunotherapy outcomes. So what do we know so far? Most of the work has been done preclinically, and the last point, which I bolded here, is the clinical work done. So thus far, we know that microbiota composition differs between patients who respond to immunotherapy versus those who don't respond, that antibiotics are a risk factor for death after initiation of immunotherapy. We also know that immunotherapy responsiveness can be transferred through fecal transfer into mice, and the response can be restored with the oral administration of key bacterial species. I think the step that really has moved this field forward in the clinical setting is certainly what we have learned from fecal microbiota transplantation from immunotherapy responders to non-responders that was able to rescue patients who had failed to immunotherapy. So just going a little bit into the details of this. This is a slide that summarizes basically the results of the two important fecal microbiota transplant trials that were published in Science in 2021. Fecal microbiota transplantation consists in the transfer of the entire gut microbiota from one host to another one, just to give you the background. In the left-hand side of your screen, on the graph on the left side, there is a phase 1 study that was led by the Israel group that basically assessed the safety and tolerability of the fecal microbiota transplantation and the reinduction of anti-PD-1 in 10 metastatic melanoma patients who were refractory to anti-PD-1. In this trial, patients receive antibiotics for 72 hours prior to the fecal microbiota transplantation. And in this study, 2 patients had partial response and 1 patient had complete response out of the 10 who underwent this treatment. If we're looking on the right side, the right graph, this is a study led by the Pittsburgh group that evaluated the safety and efficacy of responder-derived fecal microbiota transplantation and anti-PD-1 in, again, PD-1 refractory melanoma patient. The combination here was deemed safe and induced a response that was also durable in terms of microbiota perturbation in about 6 out of the 15 patients that were treated who derived what we call a clinical benefit. So not necessarily a response, but obviously stability also of the disease. Now, there are several strategies to manipulate the intestinal microbiome. And as you can see from this in this cartoon, once we move from left to right and we go into the more complex one, like fecal microbiota transplantation, basically there is an increase. Apologies. Basically, there is an increase in complexity and increase in high risk in terms of safety and scalability. And microbial consortia, who are placed in the middle here, appear to be kind of a nice balance between the 2 extremes, and therefore are or appear to be appealing compounds to bring into the clinic. Now, I did this a few months ago, so perhaps it needs to be a little bit even updated. But if we search on the clinicaltrial.gov for microbiome interventional study in cancer, we can find a large number of trials that across the globe, pretty much, are evaluating various types of probiotics and prebiotics. I left the diet out of this snapshot, otherwise it will be many slides. But only a minority are looking into immunotherapy, as you can see here highlighted in gray. If we move to fecal microbiota transplantation space, aside from the 2 published studies already presented, the list becomes shorter, as you can see. The number of patients evaluated is kind of limited somehow, but certainly there's more data with respect to fecal microbiota transplantation in combination with immunotherapy. And then if we go into the microbial consortia study that appear to be registered, these trials are all in solid tumor, in the solid tumor space, and most of them are obviously involved in immunotherapy. And we are actually leading some of this trial. So just to give you a background, we've been collaborating on the microbiome and immunotherapy kind of setting for quite some time now. We have run first observational study, which I haven't summarized here in the interest of time. And then we went ahead and started interventional study. We have done this in the advanced setting with our MET4IO study that will be shortly presented in advanced solid tumors, either naive to immunotherapy or already pre-exposed to immunotherapy. We also have used this novel microbial consortium in the locally advanced curative setting. So these are head and neck cancer patients who were receiving chemoradiation as their curative treatment, and they've been exposed to MET4. Lately, we also have been using MET4 in combination with immunotherapy in patients with melanoma, but curative disease. So those are the high-risk patients who undergo adjuvant immunotherapy. And these patients, as part of the trial, were exposed to either immunotherapy alone or in combination with MET4. So we have quite a lot of experience in using this agent. And then I'm going to turn it over to Bryan, who will present the rest of our data.
Thanks. So, what is MET4? Part of the appeal of discussing microbial consortia is some of the practical advantages over FMT, but I'm going to highlight some of these and then we'll compare some of the differences in the next slide. But it's an oral compound of human-derived organisms isolated from the stool of a healthy donor that are then cultivated ex vivo, which produces a consortium of organisms that's can be reproduced at an industrial scale even. It can be scaled up to large quantity production. It can be produced without batch-to-batch variability associated with having a bowel movement or an inter-individual differences in donors for compared to FMT, for example. And each organism can be individually assessed and characterized, including for phenotypes like antimicrobial resistance or pathogenicity or virulence. So each organism you can assess the safety of each component of the consortium. MET4 was a first-in-class consortium of taxa associated with immunotherapy response. So it selected organisms that were built into a consortium which reflected what we felt, or what the company felt was the state-of-the-art with respect to immunotherapy response associated taxa. So it's a way of building consortium ex vivo with human-derived organisms that you can scale to production for things like human trials or therapy. Next slide. And this is there's a lot on here, but some of the key differences between something like MET4 cultivated consortium and FMT include things like the composition, which can be defined explicitly. Inclusion and exclusion of organisms is an active process, so every organism that goes in has to be selected, unlike FMT, which is defined by the donor. The organisms can be individually characterized, as I mentioned, including for things like antimicrobial resistance, and there have been antimicrobial resistant organisms transferred for fecal transplant studies for other indications. The screening of the donor can be done at the level of the stool and donor, but also at the level of the organism. At the time of stool collection, screening of the pathogens can be screened, but also during the manufacturing process. It's not just the donor sample, which is the case for FMT, that has to be screened, but actually the product that's being administered can be assessed for quality and safety. It's given orally. This is variable in FMT. Increasingly, people are using pills for FMT. There's consistency, stability at room temperature. It can be taken as a pill with high tolerability, and the bacterial cells can be, the individual species can be manipulated and compared across studies, or be made available in public repositories for expansion. Next slide. The way that MET4 was designed was to replicate some of the, it has two main sort of compositional aims. One is to represent taxa or species or strains isolated from healthy human donors that are associated with immunotherapy response, but also to satisfy ecological interdependencies of species. So organisms, just like a macro ecosystem, live together. They don't tend to survive individually in complex ecosystems. And in order to satisfy some interdependencies of organisms that rely on one another to successfully grow or engraft in a host, you have to provide other organisms. So one of the aims of constructing a consortium is to satisfy those interdependencies of organisms. So they both include organisms associated with or designed to augment immunotherapy response, and also to satisfy the interdependencies of a community that enables or facilitates engraftment of those taxa into a recipient. One of the species included, Akkermansia muciniphala, for example, has been shown to be associated with response to immunotherapy. And one of the original studies published in Science documenting this relationship between immunotherapy responsiveness and the microbiome in Science 2018. Next slide. We undertook a study of MET4 compared to a placebo in individuals with solid tumors. This was, there were 3 or 4 arms in this study, but what this study allowed us to do was first assess safety by giving the consortium to immunotherapy recipients. In that context, safety is important to assess. This population is different than people, for example, with C. diff. to compare it to a control arm in individuals who are getting immunotherapy after having received so in an immunotherapy naive cohort, this was cohort B, we did have a third cohort and then actually a fourth cohort in which we provided MET4 and other clinical contexts. But the important thing here is our ability to compare MET4 to recipients of placebo in terms of, or of control arm in terms of safety and tolerability. Next slide. It's a very tolerable intervention. We saw nothing attributable to the consortium itself. This is the main burden associated with this is probably just the number of pills. These consortia have been used for other indications and a few have been published now. It's basically the main toxicity is taking the pills, but the intervention itself is very safe. We saw no severe or high-grade attributable safety events in this group, in this cohort. It was very well tolerated. So it's an easy to take and safe intervention, even in immunotherapy recipients. Next slide. And this is one of the things that we learned, which is, I think, really interesting from a future studies perspective, is that when you give organisms to a recipient, you actually see quite a lot of variability in how those organisms take or their engraftment in the recipient. So each one of these plots here, the MET4 recipients are in green and the controls are in white. Each one of these, the y-axis is a measure of how much MET4, how much of the consortium actually engrafted in the recipient. The first two plots show that there are more MET4 taxa. There's a greater proportion of MET4 taxa and more MET4 taxa in the recipients than in controls, and that these numbers actually go up. But the other notable thing here is that there's a lot of heterogeneity in these responses. Some people engrafted a lot of the MET4 consortium and some not very much at all. Next slide. One of the reasons that this may have been the case was that this was a heterogeneous cohort, and our next trial is of a much more homogeneous recipient population. So the MET4IO trial was of people receiving immunotherapy for multiple tumor types, and those patients differ from one another and may have different ecological responses to the intervention on that basis and different ecological starting points. The Promote Head and Neck trial is of head and neck cancer patients only. It's a much more homogeneous population where we would expect to see less variability from patient to patient in terms of that's based on the clinical status of the patient and the tumor type. And this will allow us to ask considerably more focused questions, both based on the ecological responses and also the clinical responses. Differences in clinical responses between tumor types are eliminated in the trial when you have only one tumor type. Next slide. The aims of this new upcoming studies to compare objective response rates between intervention groups, those that receive MET4 and those that don't. So we have clinical hypotheses, but also, and I'm going to talk about this more a little bit because this is a translational talk, we have ecological hypotheses. And one of our questions is just what happens with these consortia when you give them in this setting. Next slide. And there are lots of interesting biological questions that we can only ask in a human study, human trial like this. And some of them are very basic based on this novel form of intervention. Things like, can MET4 lead to stable microbial community composition? When we are aiming to change the intestinal microbiome, do we achieve those changes with microbiome targeting intervention? And then do we achieve changes in the host or the tumor or the clinical status based on the differences that we induce in the recipient microbiome? So there's an opportunity for us to overlay ecological outcomes and biomarkers of other types in this setting. Next slide. And this is how I think of microbiome targeting interventions and microbiome-informed therapeutics. First, we want to know what the ecology of the intervention is. What effect does the intervention have on the ecology? And that's we call pharmacoecology. How do the interventions affect the recipient microbiome composition? Pharmacomicrobiomics, which isn't the main aim of HN12, but is related, is how does the microbiome affect drug metabolism and distribution? And then microbiome-informed biomarkers, things that we wouldn't necessarily think about as being relevant in the context of cancer, but are relevant in the context of microbiome targeting intervention. So direct measures like how do you change the microbiome composition? How do you change microbiome function or transcriptomes or proteomes, but also indirect measures of the microbiome, of the effect of the microbiome. Things like how giving a gut targeting microbiome consortium affects non-target microbiomes like the mouth, skin, or elsewhere. how microbiome targeting affects stool, plasma, or urine microbial metabolites, and also how therapeutic microbes induce immune responses in the host, such as bacterial antibodies or T cell responses that may be related to tumor immunotherapy responsiveness. Next slide. So really, I think that Anna and I are really excited to be doing this project, but one of the things that I'm really interested in hearing about is what ecological, physiological, or translational questions you guys would have for a trial like this, and what questions you think are worth addressing and answering in microbiome targeting therapeutic trials in cancer going forward. Thanks very much.
Thank you very much, Brian and Anna. That was a very exciting overview of this exciting trial that you guys are conducting there. So I think we're going to get to our last speaker of the day now, who's going to give us a slightly different flavor of how you can transform an immune cold tumor to immune hot. So next we have Dr. David Uehling. Following completion of his PhD in synthetic organic chemistry at University of California, Berkeley, David did a postdoctoral fellowship with Professor Stuart Schreiber at Yale and Harvard universities. From 1989 until 2008, he worked at GSK in cardiovascular, diabetes, and oncology research. During this time, he was actively involved in identification of clinical compounds, lurtuthican for colorectal cancer and solabergon for type 2 diabetes. I'm bad with the drug names. I think that there's a special hell for people who name these drugs. Hopefully you were not one of those, David. More recently, David led preclinical teams that discovered dabrafenib, I mean, that's easier on the tongue, a drug which has been approved for the treatment of patients with unresectable melanoma who have BRAF mutations, and also BRAF mutations in non-small cell lung cancer. In January 2009, David joined the OICR Drug Discovery Program, and that's led by Dr. Rima Lawar. While at OICR, David has been involved with a variety of projects to help further the Institute's mission of translating Ontario-based discoveries into effective medicines for the treatment of cancer. So David will provide a therapeutic innovation perspective on the topic of the hot versus cold tumors. Go ahead, David.
All right, thank you very much for the introduction, and thank you to the organizers for giving me the chance to speak at this exciting series. And I just wanted to reinforce what other speakers have already alluded to, is that there's been some major groundbreaking success stories with immuno-oncology drugs, and particularly biologics. And I'm just highlighting here some of the approved drugs that are biologics that have helped transform cold tumors into hot tumors and to give impressive therapeutic responses. They include anti-PD-1 drugs, anti-PD-L1, more recently LAG-3, another speaker had highlighted this target, and those are all monoclonal antibodies, as well as bispecific antibodies, which have just started to become approved. Blincyto is one of those drugs. And then we also have CAR Ts, which are engineered human cells. And of course, with this audience, I probably don't need to belabor the exciting results of some of those drugs. But these drugs represent only a subset of the different modalities that are being pursued for anti-cancer, and this is a very full slide, I realize, but I just wanted to highlight a few of the other approaches. There's vaccines. We're starting to see there's a lot of effort. Some of the clinical responses have yet to bear out, but we do have at least one drug that's been approved. And we have recombinant cytokines such as IL-2, which have been around for a while. And of course, monoclonal antibodies and CAR T cells are the two approaches that I spoke of earlier. And we have oncolytic viruses, which now there's become a resurgence of research due to some recent approvals. And all of these drugs are biologics. And I suppose I should add, this is an incomplete table now with the previous speaker talking about the microbiome. I think that's an additional very important approach. However, among the ones that I'm showing here, with the vaccines through the oncolytic viruses, all of these have certain limitations, including difficulty in manufacturing, toxicity due to adverse immune response that's adverse events, and also some of them don't work so well in cold tumors. They only work in certain tumor types for various reasons. And so what I wanted to highlight is a last modality is small molecules, and these have their own challenges, but they also have their own attributes. And with small molecules, I think there's a lot of growing interest in this approach, as evidenced by these review articles that have appeared in the last 5 to 7 years. And before I go further, I guess I should try to define a little bit more about what a small molecule is. And so small molecules are typically drugs that are less than 1,000 Daltons. They can penetrate the cell, which is a very important attribute because of their size, and they can be dosed orally or IV. And the real attribute is that the toxicity can be managed in a convenient, more conveniently than say monoclonal antibodies, which have long half-lives, by intermittent dosing. And these drugs are usually evolved from chemical libraries starting points or natural product starting points after extensive optimization. And this is really the focus of the Drug Discovery or Therapeutic Innovation Program here at OICR. And there's really two fundamental classes of targets that small molecules can modulate. One is cell surface targets, and many of these are actually also covered by monoclonal antibodies. But because there's cell penetration properties of small molecules, there's a whole new avenue of or a whole new constellation of targets which can be directed towards, and those are intracellular targets. And that's where our group has really focused on some of these intracellular targets. But just to say that there's actually some interesting compounds that are already in the clinic that are small molecules that are targeting both cellular membrane and intracellular targets. And I just show some examples of those. This is by no means a complete list, but there's drugs that target the adenosine, which is adenosine in tumors is actually responsible for an immunosuppressive phenotype. And by having adenosine antagonists, the thought is that these molecules can reverse that phenotype and turn cold tumors into hot tumors. We've already talked about anti-PD-1 in the context of monoclonal antibodies, but very interestingly, there's small molecule drugs that actually function in the same way as monoclonal antibodies against PD-1. And some intracellular targets that have emerged are IDO and ARH, which are targeting the kyranine pathway, and basically also targeting mechanisms by which tumors lose their sensitivity to the immune system. And finally, a drug that has emerged recently is a PARP7 inhibitor, which is showing very promising preclinical results and has recently entered the clinic. I think that the jury is still out on how well any of these drugs are going to perform, but I think it's fair to say that there's been some encouraging results with some of these drugs. And I think that's really fueling the excitement for further work in the area of small molecules. And so with the OICR Drug Discovery Group, couple of targets that we've been interested in are intracellular targets, and they're MAP4K1 and CBL-B. And MAP4K1 also goes by the name of HPK1. It's a kinase which is a very druggable class of targets, and CBL-B, which is another intracellular target, which has very interesting drug-like pocket that we're beginning to learn much more about. Both of these are being pursued as CTIP projects, so we're getting generous funding from the OICR CTIP program, and we're making I think we're making very good headway. Just to speak a little bit more about HPK1, HPK1 is in a number of cells in the immune compartment, but most notably cytotoxic T cells. And essentially what it does is it dampens the signal transduction cascade between the TCR receptor and the nuclear receptors, which are involved with stimulation of the immune compartment. And in particular, it affects SLP76, which is one of the signaling partners along that cascade, and causes degradation. So essentially HPK1 knocks down the stimulation of the immune system, and it is thought that if you take an HPK1 inhibitor, what you're going to do is decrease the phosphorylation of SLP76, which is the way it transduces the signal, and increase transcription and release of various cytokines, which will then lead to stimulation of the immune system. And there is good preclinical evidence for these targets. And then shown in the top, there are a number of organizations which are interested, particularly in HPK1. And I'm showing data from another organization called Nimbus Saturn. And what they've been able to show is that with their small molecule HPK1 inhibitor, they are able to cause release of IL-2 induction in the presence of PGE2 or other immune suppressive reagents. And in an in vivo setting, in mouse models, what they show is that what they're able to show is that there is some modest single agent activity, but in particular, there's an additive or a synergistic combination effect with anti-CTLA1, anti-CTLA, or with PD-1 antagonists. And they're shown here is data from the CT26 model, but there's other models, particularly immunologically cold mouse models, which also show good efficacy with an HPK1 inhibitor. And down on the bottom is data that's very similar for the CBL-B inhibitor, where you can see again some activity with the single agent, but it's really in combination with the inhibitor of CBL-B with PD-1, where you get the profound effect both in terms of tumor volume and survival. And so many of these agents are just starting to enter the clinic, and it's unclear yet how well this data is going to translate into the clinic, but we're very optimistic. And so within our own program, we've actually established a robust drug discovery cascade where we've been able to identify compounds and subject them to an extensive cascade of testing, starting with cellular assays, and then going into pharmacokinetics, and then looking at in vivo pharmacodynamics, and then finally going into in vivo efficacy models. And so we've used these assays, and we've been able to get some very encouraging data with some lead compounds, which is just bearing fruition. But things are looking very exciting. And I just wanted to highlight that small molecules do have their challenges. And one of the big advantages of monoclonal antibodies is the fact that they can be made in exquisite selectivity for their target. Small molecules, on the other hand, often target pockets of an enzyme or a receptor, which are very similar to the pockets of other enzymes and receptors. And sometimes that's not a problem, but other times that can be a really detrimental problem if you don't see the selectivity. And in the case of HPK1, we were very concerned about a very closely related protein called GLK, and the active site of HPK1 and GLK are very similar. However, there are some subtle differences between the two, which we've been able to exploit. And through X-ray crystallography, where we actually look at how our molecules are docking into the receptor, we've been able to exploit these differences and to look at getting very selective compounds. And just shifting gears a little bit as I close out my portion of this series today, I wanted to just highlight another target that we're interested in, and it's being funded through our CTIP program, and that's ZIAP, inhibition of apoptosis protein. And we've been, these type of molecules have been known for quite some time, and as the name implies, that when you inhibit an inhibitor of apoptosis, it's a pro-apoptotic effect. But what's becoming more appreciated recently is the idea that in addition to their intrinsic cytotoxicity towards tumor cells, there's also a strong modulation of the immune compartment. And so in terms of in loss of CIAP 1 and 2, you could actually convert TNF alpha from a survival to a death signal for cancer cells. And also loss of these proteins in immune cells induces a kinase called NIK or NIC, which provides survival and co-stimulatory signals for B cells, T cells, and dendritic cells. And we're starting to take on with a company which we are collaborating with, who's really driving this, is Protaxis, working with Eric Lacasse and James Jaquith. We are actually pursuing a interesting approach where we're combining these ZIAP inhibitors with PROTACs, and the idea is that these PROTACs are actually targeting other types of molecules that may be important as tumor drivers, and there's all kinds of combinations that can be pursued. But it's a very hot space right now, the area of PROTACs, where one actually not only degrades, not only inhibits a protein, but degrades the protein of interest. And by taking a modular approach and attaching IAP ligands to ligands which bring in ubiquitin ligases, we can actually target a wide number of proteins of interest. And so we're very excited to be a part of this collaboration with Protaxis Therapeutics. So in summary, I just wanted to highlight that despite the success of biologics and small molecule immuno-oncology agents, there's a lot of opportunity in our view for the development of small molecule therapeutics. These types of drugs can complement as opposed to compete with validated approaches. And I think that there's opportunities for them as single agents, but also to help not only augment the activity of PD-1 and other very successful drugs, but perhaps even rescue drugs that haven't shown too much activity in the clinic. And through our CTIP pipelines, we've developed some nice promising projects, and we're tackling diverse target classes. And I think there's lots of opportunities down the road for additional targets. But our overall goal is to discover made in Canada, best in class small molecule drugs. And I'd like to thank our drug discovery group. So many people in our group have had a role in the projects that I've talked about that it's hard to single them out. And actually, this is an outdated photo, but it's been a real team effort. And we've also been very fortunate to work with Gil Privé and his team at UHN through doing some X-ray crystallography, as showed with HPK1. And of course, with Protaxis, Eric Lacasse and James Jaquith have been great collaborators and great partners as we pursue that approach. So with that, I'm just wrapping it up and be happy to take any questions.